What is the first thing that comes to your mind when you think about carbohydrates? Obviously, you will realize carbohydrates as a major portion of our food, right? And you might have also heard about glucose using in hospitals and for tired sportspersons. Glucose is an example of the simplest carbohydrate. If you start talking about food and carbohydrates, the list is endless. But does carbohydrates only present in foods? Now tell me, can you recognize wooden furniture and cotton clothes as carbohydrates? Yes. In fact, these fall in the category of cellulose, whose basic units are carbohydrates. Not only in the textile industry, carbohydrates also have a role in paper, lacquers, and breweries. So yes, carbohydrates are present in food, textiles, etc. Now, my question is, how do carbohydrates accommodate these diverse modifications? Well, the answer is the structures by which they are formed. In this video, we will discuss the structures of a few carbohydrates. Let us start our discussion with fructose. Fructose is a natural monosaccharide found in fruits, honey, and vegetables. You might have heard the use of honey as an instant source of energy in the Ayurvedic system of medicine. Well, let us see how fructose can be obtained. As mentioned in the previous video, fructose is obtained by the hydrolysis of sucrose, that is, reaction of sucrose with water and breaking it down into small molecules like fructose and glucose. Here, fructose belongs to the D series and is a levorotatory compound. Next, we will see the open structure of fructose. Fructose is composed of 6 carbon atoms with a keto functional group at the second carbon. If you look here, you can see keto group at the second carbon. Hence, fructose is called a ketohexose. Fructose also exists in 2 cyclic forms. You can see that these structures are obtained by the addition of a hydroxyl group at the C5 to the C double bond O group. As you can see here, this one is the C5 group which is obtained after addition of an OH group here. Thus the ring thus formed is 5-membered and is named furanose. You may also represent these anomers by Haworth structures as alpha and beta D fructofuranose. Let us now move on to the structures of disaccharides, that is, carbohydrates containing 2 monosaccharides. We will start our discussion with sucrose. As mentioned before, disaccharide sucrose is made of 2 monosaccharides, glucose and fructose. These monosaccharides are held together by a glycosidic linkage. Here you can see the glycosidic linkage formed between C1 of alpha D glucose and C2 of beta D fructose. As you can see here, alpha D glucose is connected from the C1 to the C2 of the beta D glucose through the glycosidic linkage. Now, can you tell me why sucrose is called a non-reducing sugar? You can see here the reducing groups, that is aldehydic or ketonic group in the monosaccharides of sucrose are involved in the glycosidic bond formation. Hence, sucrose is a non-reducing sugar. Note that if the aldehydic or ketonic group in the monosaccharide is free, then it falls in the category of reducing sugars. Let us see an example of this category. See the structure of yet another disaccharide, which is called maltose. The monosaccharide unit in maltose is alpha D glucose. Here, the carbon 1 of the first glucose is linked to the 4th carbon of another glucose unit. As you can see here, C1 is connected to the C4 of another alpha D glucose. Hence, a free aldehyde group can be produced at the carbon 1 of the second glucose in solution. This indicates that an aldehydic group is free, or in other words, you may say maltose is a reducing sugar. Now, let us learn about lactose. Lactose is more commonly known as milk sugar, since this disaccharide is found in milk. Here you can see the structure of lactose in detail. Lactose is composed of beta D galactose and beta D glucose. Here, the glycosidic linkage is formed between C1 of galactose and C4 of glucose, as you can see here. C1 of the beta-D galactose is connected through this glycosidic linkage to the C4 of the beta-D glucose. Similar to the structure of maltose, a free aldehyde group may be produced at the C1 of the glucose unit. Hence, lactose is also a reducing sugar. Moving on, we will look into the structure of polysaccharides. Polysaccharides contain a large number of monosaccharide units joined together by glycosidic linkages. They mainly act as food storage or structural materials. Let us see a few examples of this category. Firstly, let us discuss starch. Starch is a polymer of alpha glucose and consists of two components, amylose and amylopectin. Amylose is a water-soluble component of the starch that constitutes about 15 to 20% of starch. Chemically, it is a long unbranched chain with around 200 to thousands of alpha-D glucose units held together by glycosidic linkage between C1 and C4. As you can see here, these are an unbranched chain which is connected to C1 of one glucose with the C4 of another glucose. The other component, amylopectin, is the water-insoluble component of starch. It constitutes about 80 to 85% of starch. It is a branched chain polymer of alpha-D glucose units where the chain is formed by the C1 and C4 glycosidic linkage. As you can see here, C1 and C4 is connected through glycosidic linkage. And compared to amylose, here you can see a branching which is through the C1 and C6 glycosidic linkage. As you can see here, C1 and C6 are connected together. Yet another polysaccharide is cellulose. Cellulose is the major constituent of bacteria and plant cell walls. Structurally, cellulose is a straight chain polysaccharide. It is composed of beta-D glucose units. These glucose units are joined by glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. As you can see here, C1 of one is connected to the C4 of the other unit. Now, we will see glycogen. Carbohydrates are stored in animal bodies as glycogens. The structure of glycogen is similar to amylopectin but highly branched, as you can see here. They are present in liver, muscles and brain. Whenever the body needs glucose, enzymes break this glycogen down to glucose. I have already commented how essential carbohydrates are of life in both plants as well as in animals. Apart from the above discussed source, carbohydrates are found in the biosystem in combination with proteins and lipids. More importantly, there are aldopentoses present in nucleic acids, which are biomolecules involved in transferring hereditary information from one generation to another. We will learn about these biomolecules in the next video. Now, we will summarize whatever we have covered in this video.
Manorama Horizon. Learning made simple.